Okay, so today we are going to be talking about dimension reduction and principal component regression. I'm going to share my screen, and we have HTML as we usually do, .qmd file, and I'm going to open the notes here. And okay. All right, dimension reduction and principal component regression. Okay, we start off with a story here about dimension reduction. And this story comes from back in 2003. Now, 2003 was a few years after the first sequencing of the human genome. And microarray technology, it was new, hadn't been out very long, but it let the biologists measure expression in thousands of genes in a single experiment. First time in history that this has happened, and it held great promise, particularly for things like cancer. So one of the early studies that used this was looked at diffuse large B-cell lymphoma, which is the most common type of non-Hodgkin lymphoma. And researchers measured the expression for roughly 12,000 genes in a number of tumor samples. So what they were trying to determine, to figure out, was a problem that had confounded them for some time, where you could look at two tumor cells under a microscope. They would look exactly the same, but one of these people would have, the two people or the two groups of people would have dramatically different rates of survival. And so the hope was that, okay, we're going to sequence these genes here, 12,000 genes, and figure out, what's going on here. But when they got the data, it's this huge matrix of data, and they're like, well, what the heck are we going to do with this? We have 12,000 measurements and a number of samples. What the heck? So it's just too much data for them to consider at once. Now people do this kind of thing all the time. It's more standard, but at first it was like, geez, what are we going to do with this? So the breakthrough wasn't, one particular gene. It ended up being a different way of looking at the data. Here, they did dimension reduction and were able to use clustering, what we talked about yesterday, to see a small number of groups of genes that rose together and fell together at the same time. And then when they looked at these genes among the groups of people, they were able to see that, okay, if you are in this particular group, you tend to have much better 5-year survival rates than in the other group. And it really fundamentally changed the way that lymphoma is classified and treated. This is now one of the first things that they look for. Okay, which type of lymphoma? Previously, you could not tell any difference in the tumors. With the new genetic data, it made a big difference. So the significance of this goes, well beyond lymphoma. It just is a different way of looking at this type of high dimensional data that we've been talking about with dimension reduction. It's not only a genetics problem. It appears anytime you have a lot of correlated variables and you want to have some way to summarize them. So some examples here. Dozens of health indicators from a clinical exam are the example we'll use. We'll go over this. Hundreds of survey items measuring similar constructs, environmental exposures measured at many time locations. Another one is economic indicators, global economic indicators on a whole bunch of data, whole bunch of countries, how do we summarize that? So some of the issues when we have many correlated variables, number one, visualization. We can't plot 20 variables at the same time. We can't see patterns just by plotting. So when we want to do exploratory data analysis, and that's when we're just exploring the data, just exploring the space, having tons and tons of variables is a detriment to that. Collinearity. Correlated predictors will destabilize regression coefficients, something that we'll talk about later. But correlated predictors, they rise and fall together. And so when you're looking at the relationship between two variables and one of them has other variables correlated with it, it's difficult to know, is this the variable I'm looking at, or is it the other variable that's highly correlated with this, right? So that's another issue that comes up in visualization. And again, we'll see this in regression, some of the issues that these correlated predictors can have. And lastly, overfitting. So when we put a whole bunch of variables in a model, we get to that overfitting space that we talked about last week, where we have a model that memorizes the noise versus predicting the signal. So dimension reduction is a way to create a smaller set of new variables that summarize the original ones and can address all these issues. So there's two kinds of dimension reduction. First one is feature selection. We're not going to talk about feature selection today. It was on the original list of topics, so I'll just kind of briefly mention what it is. So feature selection is when you keep a subset of original variables and you discard the rest. So if we have 200 variables to start with, after feature selection, we may build it down to 200 maybe. We were doing something like that in the previous days when we were running DNA-seq. The difference between, for example, LASSO and DNA-seq. DNA-seq is each variable is analyzed separately. When you do LASSO, all the variables are analyzed at the same time. It's one model that does that. So LASSO is a way to fit a regression model when you have more predictors than observations. So you wouldn't fit it in the situations that we were, where you have 8 samples and 20,000 measurements. That's too much. You need more than 8 observations to run a LASSO. But you can do it in other situations. So this is something that I work on a lot, and we use a lot in genetic analysis. We use it in neuroimaging analysis as well, when we have predictors that have very, very high P. But it's a big topic, and we can't just spend half the day on it. So if you're interested in LASSO or some of those feature selection methods, I encourage you to dive into those. What we will talk about today is feature extraction, where we're going to transform the original variables into a new, smaller set of variables. The new variables are going to be defined by the original variables. Sometimes they're going to be linear combinations of those original variables. Example that we're going to go through is principal component analysis, where the new variables, the principal component scores, are weighted sums of the original variables that we have. The individual variables lose their original meaning. So these combinations, they don't have the original meaning, but some of the patterns we can view across them, they are going to help us with our modeling of the data. So that's what we'll talk about today when we go into this principal component analysis. Okay, so before we talk about principal component analysis, I want to step back and look at the bigger picture of dimension reduction and the different classes of dimension reduction methods. Today, we're going to talk about PCA. There are many, many other dimension reduction methods. For example, TSNE, UMAP. Those of you in the biology field may have heard of these. These are other common dimension reduction methods. So let's take a step back and kind of put dimension reduction methods into some classes. And here I'm going to separate these into global methods and local methods. So we can talk a little bit here about what the difference between these global and local methods are. So global methods, they preserve the overall variance structure. The transformation is linear, and importantly, it's deterministic, which means we can figure it out for a new observation. And we can apply it to new data. Interpret coefficients, we can do that on new variables. We can project new observations in the same way. Local methods are not as, it's not as clear how we go from the big data to the small data. So the piping from this huge data set to this small number of variables is not as clear as it is with something with PCA. But what local methods are very good at is they preserve neighborhood relationships between nearby points. The variables we end up with, they don't really have any meaning to them. And you can't easily project new data or interpret regression coefficients or any of these type of things. Local methods are really used for exploratory and visualization. We can also do that with global methods, but then in addition, we can do regression and prediction and inference with these. Okay, so I have a good analogy of local or global methods. And let's suppose that we walk into a party and we want to draw a diagram representing the people in the room in terms of their personalities, for example. And we want to use this maybe to see who is friends with whom or something like that. Okay, so global method versus a local method. A global method would have, for example, it would have these people in the room take some personality assessment, maybe. So maybe it has a measure of how introverted you are. So we find out how introverted every person is, and we get some number based on that. And then we find out how funny the person is. So, sense of humor, we get a measurement of that. And then we tell everybody, okay, use your introvert measurement and your sense of humor measurement, and that's where you're going to go in the room. Okay, people who have are low on the introvert and high on the sense of humor, they'll go in one corner, and so on and so forth. And then we kind of, okay, we have this spread of everyone in the room. Nice thing about that is when a new person walks into the room, we can put them where they need to go. We can say, all right, we have these assessments. How introverted are you? Tell us about your sense of humor. We know where you're going to go in the room. The bad thing about that is people who are actually great friends are going to be separated in this room. They're not going to be close to each other in any particular way. All right, let's keep that in mind, and then let's look at how a local method might do this. So a local method would say it would work completely differently. Instead of getting these assessments on people, would say, all right, everyone, just go stand by your friends. Okay. Go stand, look for your friends in the room, go stand by them, and that's where you're going to be in the room. In this local method, what we would find is that we end up with groups like, okay, these people are friends because they work together. This group over here, they all went to college together, and this group over here, they're friends with the host or something like that. So we end up with these like cliques of people in the local method where people who are friends are actually right next to each other. So that is a good thing about local methods and that's what I mean, they preserve neighborhood relationships. So they preserve these relationships among the people. Um, However, the axis where they are in the room has no meaning at all. And we could have maybe the work friends and the college friends, they could end up being near each other, and that doesn't mean anything whatsoever. The fact that the work friends and the college friends are close to one another. So the where things are on the X and Y axis really doesn't have any bearing on the actual personalities or anything like that. Also, if a new person walks into the room, we can't really just put them anywhere. We would pretty much have to do the whole analysis over again. We would say, okay, everyone come back. All right, get with your friends again. We got this new person. We got to put them somewhere. Now, in practice, we could say, hey, who are you friends with? But you can see that we can't really do that with data. We can't ask data, hey, who are you friends with? We have to have a deterministic way of mapping a new observation onto these new variables that we created. So global methods that we'll talk about here, PCA, it's deterministic. We can have some interpretations, so these new variables that we're creating, there are going to have some interpretations. We can, if we get a new observation, we know where to put them in terms of the room. They're not going to preserve any local relationships, though. The local methods, it's a little more unclear how we get from the big data to the small data, but they are going to preserve local relationships better. They're going to do that at the expense of not being able to project a new individual or new observation to where they should be in the data. So you will see local methods used a lot in the genetic biological setting, but you'll see global methods applied there as well. So that's just kind of all right. Let's step back and look at dimension reduction in a bigger umbrella. Put these different methods into different classes, and now we'll continue the rest of the day. We'll focus on principal components analysis. What does it do? What does it mean? What does it give us? And how can we use those results in downstream analysis? Okay, so what is principal component analysis? What it does is it creates a new set of variables. These are called the principal component. Each principal component is a weighted linear combination of the original variables we get it. The principal components have a few properties to them. Number one, they are uncorrelated with each other. It actually goes further. They are orthogonal to one another. So one principal component has a correlation exactly equal to 0 with The first principal component has a correlation exactly equal to 0 with the second principal component. The third principal component has a correlation exactly equal to 0 with the first and the second. So they are completely uncorrelated. The correlation is set to be exactly equal to 0. So each principal component explains something that the principal components before it did not explain whatsoever. All right, that's the first property of principal components. The second one, they are ordered by how much variance they explain. The first principal component captures as much variability as it can. The second principal component captures as much variability as it can while its correlation is exactly equal to 0 with the first principal component. The third, same thing. Capture as much variability, but it has to be perfectly uncorrelated with the first two, so on and so forth. So you can think of it as a rotating coordinate system to align with the directions of the greatest spread in the data. So we're going to look at this with an example here. This is just a toy data set that is, I feel like it's useful to think about what is going on with principal components. It's from the United States Department of Agriculture. It contains the nutritional content of one serving of a whole bunch of different food items. Nutritional content, we just have fat, protein, fiber, and vitamin C, just those 4 variables that we'll be considering. We're going to run a principal component analysis on these data. Now, it's not a situation that we would usually use principal components on because we only have 4 variables, but it is something where we can get nice interpretations of what the principal components are doing. So after we run a principal component, the output that we get are the loadings. So each principal component, remember, is a linear combination of the variables we give it. So here we have 4 variables. Principal component 1 will be a linear combination of fat, protein, fiber, vitamin C. The loadings are the coefficients that determine that linear combination. A large loading, so positive or negative, means that variable is influential in that particular component. Large loadings will help us interpret what the principal component is representing. So we'll get these loadings. So the loadings are going to be information about the variables. The scores are going to be information about the observations. So the loadings will tell us what's going on with fat, protein, fiber, and vitamin C. The scores will tell us, hey, if we look at kale or parsley, what kind of score did it have for principal component 1, or principal component 2, or principal component 3, or principal component 4? These are what we would use downstream in a regression analysis, clustering, or visualization. We've already looked at these because we've done principal components a few times, and we've looked at the scores among the subjects or among the genetic components. Eigenvalues and variance explained. These are information that are going to help us determine how many principal components we should keep. Now, I will say, usually when you're using this for visualization, people keep 2, okay? But when we're doing a downstream analysis, it's a bigger question of how many do we need to keep to ensure that we've explained the data well enough. And hey, even if most people usually keep 2, hey, should we keep 3 or 4 or something like that? Okay, so how do we use these eigenvalues and variance explained to determine how many components we should keep? So what we want to do is we want to explain most of the variation. So we want to explain most of the variation in the data given a particular number of principal components. The scree plot, what this plots is usually the percent of data accounted for by the principal components. So the first principal component is always going to explain the most variation. So here you can see it explains 45% of the variation. So what that tells us, if we just look at one principal component, that explains 45% of the variation in this nutrition data that we're looking at. So there's only 55% of variation left after we take that principal component out. Here we see the second principal component explains about 25% of the variation. So that's, again, that's a lot. So now when we combine the 45 and the 25, we have 70% of the variation in the data is explained by just two variables. We started with four, just two of them can explain 75% of the variation. And the elbow method is, yeah, we look for an elbow in this scree plot because once we hit that elbow, the rest of them aren't explaining as much variation. So here, if we're using the elbow method, we'd say, all right, we're just going to use two components. So that's one way to determine how many principal components you should keep. The next way is to use the cumulative variance method. So that's when you do that beforehand, you say, I want to explain 80% of the variance, and the number of components you'd use would be so that you have over 80% of the variance. Here, that would give us the first three principal components. That really gets us above that 80% threshold. Okay, so we have the scree plot method with the elbow. The cumulative variance is another way to determine how many principal components to keep. And then finally, we have Kaiser's rule, which says keep all the principal components that have an eigenvalue greater than 1. So it's a very simple method, deterministic, widely used. There's no correct answer here. So I'm just giving you the different methods that are out there. Any questions on that? Yeah, I think it gets more fun when we start talking about the loadings and how to interpret those. So each loading is the correlation between the original variable and the principal component when the data have been standardized. So if we see a large positive loading, that variable increases a particular principal component we're talking about. Let's actually just go to the loadings here. I think it's going to be easier after we see that. So these are the loadings for the nutritional content data. Principal component 1, principal component 2, principal component 3, principal component 4. There are only 4 variables in this data set, so we can only make 4 principal components. The maximum number of principal components is going to be the minimum of the number of variables we have and the number of observations that we have. If we only had 2 observations, we could only make 2 principal components, for example. But here we see the first 4 principal components, and let's talk about the loadings here. So the first principal component, which again explained about 45% of the data, it has a negative correlation with fat, a negative correlation with protein, positive correlation with fiber, and a positive correlation with vitamin C. So foods that are high in fat, high in protein, low in fiber, low in vitamin C are going to have large negative principal component 1 scores. Foods that are low in fat, low in protein, high in fiber, high in vitamin C are going to have high principal component scores. Does anybody dare a guess at what a food, for example, name a food that would have a low principal component 1 score, or a type of food that's going to have a low principal component 1 score? You can unmute yourself, put in beef. May, great job. All right, beef, high in fat, high in protein, low in fiber, low in vitamin C. Chad, very good. So fruits, vegetables, they are going to have a high principal component 1 score. They're low in fat, low in protein, high in fiber, high in vitamin C. So what this is telling us is that most of the variance in this data, first thing it's going to do, these principal components, it's going to separate things into meats versus fruits and vegetables, that type of thing. That is kind of the major differentiator between these variables that are in the data. Okay, so then we go to the second principal component, PC2. PC2, a little harder to determine. We got a figure here. I got to remember what these things are. And this one, I actually I don't have a good reasoning for this one as much, but so principal component 2, remember, has to have a correlation exactly equal to 0 with principal component 1, has to be orthogonal, so completely new information. So principal component 2, high correlation with fat, high correlation with vitamin C. So foods that are high in vitamin C, high in fat are going to have high scores. Protein and fiber are not really related to principal component 2. So commonly, if the loading is less than 0.4, you'll kind of ignore it. I don't know if anyone has a good categorization of principal component 2. I'd love to hear it, but I've looked at this one a few times. I don't really have anything great for that. Principal component 4 is almost the opposite. So protein and fiber are really what it's looking at. And principal component 3, we're looking at high fat, high fiber, low protein, low vitamin C would have high scores. But I think you get the major component here. So large positive loading, variable increases the principal component scores. Large negative loading decreases the principal component score. Near 0, the variable isn't really contributing anything to the principal component. So we're going to look at this with NHANES. PC1 might have a large loading on age, systolic blood pressure, total cholesterol. It's like a cardiovascular aging component. Principal component 2, okay, it's BMI, systolic blood pressure. All right, this is a metabolic risk component. So this is something that the first thing to look at from a principal component analysis is the loadings that you get on the particular variables. We'll go back to to that nutrition example in a little bit, but let's go to our .qmd file and take a look at our example here for the day. Okay, so loading all the packages, and again, resolving some namespace conflicts that I talked about in the lab yesterday. Saying, all right, use select from Dipler, use filter from Dipler. Those can have some conflicts to them. So here we're going to use the NHANES data set. It's just one that I chose to take a look at this. It does not end up working out with the most interesting results, but well, I think there's some things there. So this, as you probably know, a large cross-sectional survey of US adults with measurements on BMI, blood pressure, cholesterol, pulse, sleep, income, alcohol use. These variables are correlated in biologically meaningful ways. They all measure health in some fashion. So it's a good example for principal component analysis, and later we'll use it for principal component regression. So first, let's take a look at the data. This is the variables that we're going to be focusing on: age, BMI, systolic blood pressure, diastolic blood pressure, total cholesterol, direct cholesterol, pulse, sleep hours per night, income, and alcohol per day. We're going to only keep variables that have non-zero that have complete observations. We get a little over 4,000 observations on 10 variables. Here's a quick look at the data, removing some of the categorical variables: age, BMI, systolic and diastolic blood pressure, total cholesterol, direct cholesterol, pulse, sleep hours per night, income, and alcohol. Several of the variables are right skewed, so we're going to log transform those before standardizing. This is just going to make the principal component analysis works a little better on data that have a bell-shaped distribution. Not required, just works a little better. One thing that we do want to do when we're performing PCA is we want to scale the data. Why is that? Well, we are, first principal component that we get, it's going to explain most of the variation. Well, if one of our variables has way more variation than the others, it's going to say, well, I'm going to explain that variable. So if we scale it, then all the variables have the same variance, so it gets rid of that issue. Any questions so far? No? Okay, good. All right, so let's run PCA on this scaled version of the data. I'm saying don't center and scale because we've already centered and scaled this data. First thing we'll look at is the scree plot and the plot of the variance explained. So here is some code to calculate the variance explained for each variable. Here we have the cumulative variance explained, putting that into a tibble and then plotting that. So on the left-hand side, we see the variance explained for each principal component. On the right-hand side, we see the cumulative amount of variance explained. So if we're using the elbow method here, maybe we would use 5 principal components. Seems to be some diminishing returns after that, or maybe even 4 principal components, some diminishing returns after that part. If we're using the cumulative variance explained, if we wanted to explain 90% of the variance, we would use 8 principal components. I did not look at the here. I don't show the eigenvalues, but we could look at those and see how many are greater than 1. Mostly for this first part, we're going to look at the first 2 principal components and see what we get with those. And this is just that data in a tabular form. Yeah, so if we want to explain 80% of the variance, we would use 7, 90, we would use 8. Okay, let's look at the loadings. Okay, so these are the loadings for the first 2 principal components. And you can see that where we get these, PCA res $rotation. And here we're just doing some fancy plotting of these. All right, we have PCA1, we have PCA2 that we can take a look at here. So principal component one, large positive loading for alcohol, a large negative loading for age, and then relatively large for systolic blood pressure, maybe total cholesterol in there a little bit. Everything else is small. Really, even total cholesterol does not have that large of a loading. For principal component two, we get large positives loadings for BMI, diastolic blood pressure, systolic blood pressure, negative loading for direct cholesterol. We can see the values of the loadings here. So if we're using that 0.4 cutoff, really age and even alcohol is not above that 0.4. It's above the 0.3 mark. But the PC1 seems to correspond to younger participants who drink more. I think those are kind of some of the big components of that. So if you're younger, lower value for age, higher principal component one. If you drink more, higher for principal component one, and then systolic blood pressure, lower on that, higher for principal component one. And if you have a low PC1 score, you're going to be older, higher blood pressure and cholesterol, less alcohol. So not, as I said, not the clearest interpretation there. I mean, they don't always have the most clear interpretation. That's not their goal. The goal of PCA is to explain the most variance in the data. For principal component two, we have BMI, systolic, and diastolic blood pressure. They have the largest positive loadings. We see the three of those going down there. The HDL has the largest, the direct cholesterol, excuse me, has the largest negative loading. So it's almost like a metabolic syndrome axis. PCA3, negative loadings for age and BMI, positive loadings for direct cholesterol, pulse, and total cholesterol, also a positive loading for alcohol. So, interpreting PCs is part art, part science. The signs and the loadings are arbitrary. We could flip these signs and get the same thing. So it's really only the pattern of which variables are loading together, their relative magnitude that carries some information. Again, so sometimes you can look at these and you can see some clear patterns. Other times, you can't. We saw this with the nutrition example as well. So looking at the loadings is a important first step in your PC analysis. Then, of course, we commonly will display the loadings graphically. A biplot is a common way to do this, where you show the observations, points, and sometimes the variables as well on the same plot. The figures that have the variables, those will be arrows. It'll show what direction the original variable is related to in terms of the PC. Two observations with similar scores on both PCs, we'll see them lying close together. Let's look at this plot first. So this one just looks at the first and second principal components in terms of with the labels for the foods that we have. So the first PCA you can see, as we said, it clearly separates things into, meat, fish on the negative for PCA1, while fruits, vegetables are positive for PCA1. Actually, it doesn't look like we have many fruits on here. It looks more like vegetables and legumes type thing. PCA2, which is a little harder to differentiate. PCA2 was related to fat and vitamin C. So variables up here, they have high fat or high vitamin C or both. And I think it really is they have high fat or they have high vitamin C. So it's not really a relationship between fat and vitamin C so much as that it's really picking out those that have high fat and those that have vitamin C. And so for the meat over here, you see that PCA2 somewhat separates things into the fish versus the land animals. And PCA2 on this side separates things from the vegetables more versus the soybeans, peas, yams, carrots. I guess these are leafy vegetables versus root vegetables and legumes and garlic. I don't even know what garlic is. It's some kind of root, right? So separating things more into that. So that's interesting. So PCA2 really seems to be kind of separating the data, but really by PCA1. So it's really taking this side of PCA1, it's doing one thing. This side of PCA1, it seems to be doing something else. So at least that gives us something. Let's take a look at this for the NHANES data. So how we can do a biplot is this FVIS PCA biplot. This will give more information than the one I just showed. So here the dots are the participants in the sample. We see a dot right here. This person has a very large PCA1, very large PCA2, while as this person here, low PCA1, high PCA2, and so on and so forth. The dots are, the colors are, and the points are actually there. So the triangles for the males and the circles for the females, and we have different colors for them as well. We're not labeling these because, it's not really all that informative. And the circles here look at, all right, the average and spread among males, the average and spread among females. So males, the PCA1 score is maybe on average a little bit higher than for females, not too much though. For females, the PCA2 score is a little bit lower than for males. Also, females seem to have more variability in their PCA1 and PCA2 scores. So we have that individual side of the biplot, but what the biplot here shows that the previous plot did not show is the actual loadings for the variables. So we can see that alcohol has relatively high positive loading for PCA1. Diastolic blood pressure, relatively high loading for PCA2. Direct cholesterol, positive loading, or excuse me, negative loading for both PCs. Age, large negative loading for PCA1, nothing for PCA2. So biplot's going to give us a whole bunch of information in terms of the relationship of the variables and the relationship. We can see where the observations are, which is meaningful when we have some labels that we want to look at for the observations. I was going to pull up the PC plot we had in previous days. There's a few of these that we looked at. I think we did this with the observations. Where's that? Gosh, I'm having a hard time. I think this was actually the first day that we looked at this. Sorry. Yeah. Okay. So this is the Facila data set where we looked at PCA1 versus PCA2. And here you can see the first two PCAs explain 86% of the variance. That's a ton. So this data set has like thousands of variables, and we can just use two and explain 86% of the data. That's a ton. And when we plotted this, we saw clear relationship with treated and untreated, clear relationship with library type. So this is, now we're not going to plot the scores for all the variables or anything like that, because that would be way too much. We also looked at this for the airway data set. Excuse me, that opened up the wrong one there. Yeah, so this was it for the airway data set. PCA1 explained 41% of the data, PCA2 explained 26% of the data, so altogether 67%, about 2/3. PCA1, clear relationship with treatment. PCA2 had a strong relationship with the cell type. So these biplots, or these plots of the data with the by the PCAs are very popular. Any questions on that? Okay. Let's look at the score. This is just another one by age group. So we see the age groups into 20 to 40, 40 to 60, 60 to 80. So PCA1 was related to age, so there's a clear relationship there. The older age groups are lower on PCA1. Not much relationship with PCA2. We know age does not have a relationship with PCA2, but some of the other variables related to age might. So that's another. So you can look at the relationship between any of these, any grouping variable. This is a nice package to take a look at how it's related to the PC scores that we could be interested in. So some important notes on PCA. Always scale your variables first. We want to use, or you can use scale equals true in the principal component function. Again, principal components is trying to estimate most of the variance. If you have a variable that has more variance, the PCs are going to be disproportionately weighted towards that variable. We don't want to do that. We want to give all the variables the same chance. If a variable has a tiny scale or a huge scale, we want to put them on equal footing. Principal components is sensitive to outliers, so a single extreme observation can pull the PC toward itself. That is one of the reasons why we log transform some of those variables when we started our analysis. With some of the variables that we had: total cholesterol, direct cholesterol, sleep, income, alcohol. So if we did not log transform those variables, we would have had some outliers that would have disproportionately impacted the PCAs that we had. So it's going to be sensitive to those, and we're going to see you're going to see an impact. So that's why we like the variables to have a more normal distribution before we analyze it. It's not required, it's not like an assumption or anything that they're normally distributed, but it is something that PCA works a little bit better when we have that because it doesn't have the outliers. PCA is unsupervised. So the principal components that we get back, they're not necessarily related to anything. And we saw this a little bit in this example. Not really great interpretations of the PCAs 1 and 2, maybe some for PCA 1, maybe some cardiovascular aging in some way related to age and alcohol and some other things. PCA 2, some kind of metabolic syndrome. Again, I'm just kind of making things up here. So the scores that we get in the end are not necessarily related to any outcome at all. They are just designed to explain the most variance in the X variables that we have. So this is a big difference between going back to feature selection and feature extraction. Feature selection, you end up with a subset of variables that are related to some outcome of interest. So those are going to be particularly chosen because of their relationship with some binary or continuous or survival type outcome. The dementia reduction methods are not necessarily related to anything. So in theory, you would think that they are, but they're not necessarily related. So one example of PCA that's kind of useful is when you have economic data on countries. So let's say we have 100 economic measurements on different countries in the world, and we want to do a PCA to explain those 100 economic measurements. Now, what we would think is that, all right, we have 100 economic measurements, but there really are maybe 5 to 6 latent factors that are really important. Okay, 5 to 6 latent factors that are really driving all these economic measurements that we get. All right, how good is the economy? How much do they trade? I don't know, and then some others. So then we would think the PCAs should pick up those latent factors that are really driving all the data. So in that situation, the PCAs we get, they should represent something. They should represent these latent factors that are driving the data. That's kind of the thought process behind them. But it's not necessarily that those latent factors are there. We don't know that. We can't test that. And they may not come up when we do a particular analysis. Where PCA was used a lot originally, back in the 80s and 90s, was in testing, like IQ testing. And the thought process there was, okay, we have all these measurements of IQ, of intelligence, but there's really only a few latent factors that are driving everything. How good you are with math, how good you are with reading and comprehending text, writing text, So there's a few latent factors that are really driving everything. And the PCAs, they should be picking up those latent factors. So those are the situations where PCA is really going to work well. And again, this is totally different than the local methods. There's no latent factors there or anything. They're just trying to keep local data together. Another nice thing is we can project new data. So if we have a new subject in the NHANES data set, we can figure out what their PCA 1, 2, 3, and 4 scores are going to be. If we have a new food in the in our nutrition data set? Well, all right, for the new food, give me its fat, protein, fiber, and vitamin C. I'll tell you its score on PCA 1, 2, 3, and 4. So that's a very nice thing about PCs is that we can predict what they are for new data. We can't do that with local methods as I described before. Okay, so again, I talked about this already, but we already used PCA this week when we used it in day 11. VST PCA, we actually used it on day 13 on those gene expression data. Genomics, it's going to serve three main purposes: quality control, do the samples cluster by treatment. So it's just a good initial check of, is there some relationship here? Do we pick it up? Does it very obviously pick up? Here, I think the other thing that I thought was important about it was that we could look at some other factors here, library type for the lab. In the airway data set, we could look at cell line and see, all right, is there some need for covariate adjustment? And so if we, for example, if we saw batch effects in the data that we looked at, that would be a clear sign that, well, these were run in different batches, and batches ended up having an impact. For those ones, we had that single read or double end read in one, and we had the cell type in the other. So quality control visualization reduces 20,000 genes to 2. So particularly for visualizations, I think this is something where if your goal is really visualization, then you can consider local methods as well. The TSNE, the UMAP, those are really cool methods to use, and they can pick up things that PCA will completely miss. So if you're really doing visualization, I think those are interesting methods. Just remember some of those notes about them. We'll take a break here, and when we'll come back, we'll talk about using the results from principal components in a regression model. This is a nice thing that we can do. You wouldn't do that with the local methods, but this is something that we can do to simplify a regression analysis and maybe end up with something that is that works a little nicer than just using the original data. Great. So I got 11:05 here. Let's come back in 15 minutes. We'll regroup at 11:20, and we'll continue on with our exploration of principal components analysis, in particular, talking about principal components regression. Great. I'll see you in about 15 minutes. All right. Thank you, Elena. Let's continue on. Okay, so far we have talked extensively about PCA analysis, and now I want to talk about one of the ways to use it, which is in regression. So when we talked about linear regression on day 6, I mentioned how linear regression is unstable when you have certain predictors that are highly correlated with one another. For example, like maybe diastolic and systolic blood pressure. We had another one that came up that way. And when this happens, when you have variables that are highly related to one another, the coefficients become unstable. Standard errors are going to increase a lot. Coefficients signs can flip. So you can have a variable where you're like, oh, I know this has a positive effect, but when you do linear regression, it has a negative effect. and why that negative effect is coming up is because of that variable's relationship with another variable or variables in the data. So for example, if we're predicting blood pressure from a clinical panel that includes BMI, weight, waist circumference, and cholesterol, all highly correlated variables, no single coefficient can become interpretable because the interpretation is what's the change in blood pressure for a 1 unit increase in BMI, holding weight, waist circumference, and cholesterol constant. Doesn't really become interpretable because you can't really do that because they're so highly related here. PCA solves this problem by replacing the correlated predictors with a smaller set of principal component scores that are all completely unrelated. There's no correlation at all among these principal component scores. So it's a good way to do that. Okay, so how do we do this? We do this in 2 steps. First, we run the PCA on the predictor matrix X. We keep the first R principal components. Then we would regress our outcome Y on the R principal component scores instead of the original predictors. The PC scores are uncorrelated by construction, so there's no collinearity, so you don't have to consider, you don't have to worry about any of that. The regression coefficients on the PCs are interpretable. So if you have a coefficient gamma k, it's the change in y for a 1 unit increase in the kth principal component. And if this all works out well, we can connect those principal components to latent constructs in the data like cardiovascular aging or metabolic health. So we can say, well, for a 1 unit change in the cardiovascular aging principal component, this is what we expect for the change in the outcome. So let's go to our NHANES data set. And take a look at that. Okay, we have all these variables that we're keeping: age, BMI, blood pressure, all those types of things. And here we're looking at the correlation matrix between these variables. We can visualize it using the core plot package a little better. Really, there's not any tremendously high correlations here that I would be concerned about. That's what I was saying. This example did not end up turning out perfectly for all these things, but it's some relationship between direct cholesterol and BMI, some relationship between alcohol and age, some negative correlation there. Everything else, correlations are pretty small. Now, we can have all small correlations and still have collinearity be a problem because collinearity is there whenever we have a variable that can be explained by a combination of other variables. So it doesn't have to be just one variable is explained by one other variable. It could be that BMI is predicted very well through a combination of cholesterol, sleep, pulse, something like that, maybe. So looking at this, I would say, well, I'm not really worried about collinearity at all based on this. Still could be a problem, but likely we're going to be okay with this data set. So here we're doing standard OLS, and there are some large standard errors for the cholesterol variables for the sleep hours per night. This is likely due to the scale of these variables, I would say, more than collinearity. All the signs are in the direction that we interested. Again, our outcome here is systolic blood pressure. So diastolic blood pressure, positive sign, age, BMI, total cholesterol, direct cholesterol, all positive signs. Pulse, sleep, uh, income, all negative signs, alcohol, positive signs. All those are how we would expect them to be. So overall, it doesn't appear that this is going to have too many issues. So how we would fix this if there was some issues is to do principal components on this. Here, scaling the matrix, doing the principal component, the proportion of variance we see here. So the first 1, 2, 3, 4, 5 variables are going to explain at least 75% of the variance. So it's a reasonable choice here to use. So I'm saying R equals 5, and this PCA underscore pred, that's my principal component object, $X, that's going to give us the principal component scores. So, and I'm asking for the first R, here the first 5 scores, principal component scores for all subjects in the data. Here I'm just renaming it PC1 through PC5. And that is added. We have the scores data. We have Y, which is going to be our systolic blood pressure. And then we're fitting that and we see the relationship here. So the first principal component, negative relationship with the outcome. That first principal component, if we go back to the interpretation, was related to age and alcohol use. So it seems to have, when you have a higher principal component 1, you seem to have lower systolic blood pressure. The second one, which was the metabolic risk type 1, highly significant coefficient, highly significant, and with the systolic blood pressure, large coefficient estimate. And then PCs 3 and 4, also highly significant, strong relationship with the outcome. So this is all looking pretty good. PCA 5, things drop off there. And we could go on. So principal components 6, 7, and 8 and 9 we're not using here. Those explain less variation in the data than the principal components we're including. But that doesn't mean that they're not related to the outcome. I mean, principal component 9, the last one, well, that explains, the smallest amount of data. It could be very important for predicting the outcome. There's nothing to say that. So that sort of me trying to accentuate the fact that the principal components we get aren't necessarily related to our outcome in any particular way. We didn't tell the principal component analysis about systolic blood pressure. We didn't tell it we were looking at systolic blood pressure. We just gave it the data and said summarize it. The R squared here is 18.3%. When we use the full data, we got 32.3%. So we are missing a chunk of the explainable variation. That's not like a small amount, 18 versus 32, about 14% of the data that this is missing. So the PCAs that we eliminated, they seem like they're important still. So how many PCs did we choose? I chose 5 there as 75% of the data, but we may want to choose more. So the right number of PC is the one that gives the best predictive performance, which is not necessarily the most variance in X, as I said. The 2 criteria can disagree. A PC can explain little of variance in X, be highly associated with Y. And a PC that explains a lot of variance in X might be might have no relationship to Y. So one way we can do this in practice is to do cross-validation. We talked about this last week, where we'll do a principal component on the training data. We'll have test data set aside, and we'll fit a linear regression on the training data. And then for the test data, we'll predict the principal components using the model from the training data, and then predict the outcome based on the model from the training data, and we see how well it predicts. This function, PCR in R, is going to have this capability where it can do cross-validation to help us select the number of principal components to choose. And I think here the results are pretty interesting. So here we're going to use this function, okay, PCR. This is going to do everything for us all at once. So I give it the outcome, I give it all the variables that we are using. I'm telling it to scale it because this data I'm giving has not been scaled. And the validation here is going to be cross-validation. So that is going to help us choose the best number of principal components using the idea of cross-validation. So here we see the error by the number of principal components. And typically what you would use is you would find the number of principal components that have the lowest predictive error. Here, it gets us all the way back to 9. 9 is the maximum number of principal components we can have because we only have 9 variables. And really, there's no point in doing a principal component analysis with 9 variables because we're not summarizing the data at all. We're getting different variables, and maybe to you, those variables are more meaningful than the original variables, but likely in that case, I would just, hey, let's just use the original data. So the optimal number of components ends up being 9, which is going to give us a model that is equivalent to the full model that we originally started with. It'll have the same R squared, the same residual standard error, same overall p-value, different coefficients because they'll be the coefficients for the principal components, but in all essence, the model is going to be the same. We can see here R squareds are going to be the same between the best fitting PC principal component regression model and the model with all predictors because they're the same. We can back transform, so we can use the coefficients from the PCR fit to get coefficients for the original variables. So this is just some code to do that. Any question on the principal components regression piece? Good. Okay, so some of the advantages of principal component regression. It eliminates collinearity, reduces overfitting. I think this is the main one here, is that it works when we have more variables than our sample size is. So we could have 20,000 variables and 100 people. We can do principal components on those 20,000 variables, get them down to maybe 10 principal components, and then we can predict our outcome that we're interested in. So that's kind of the main advantage and the main place that I would use it. It's also, we can apply this to new data. That's a great thing about principal components. Some of the limitations, the principal components don't necessarily have anything to do with our outcome. So we just did this with systolic blood pressure. The principal component analysis doesn't know what our outcome is. So it's just trying to summarize the X variables. It's not really maximizing the predictive nature of PCA1 with Y in any sense. Interpretation is harder than on the original variables. Um, so that's going to be a limitation of it. The number of PCs, how many we're going to choose. So that's a tuning parameter, something we have to deal with using, say, cross-validation. And again, PCA is an unsupervised technique. It ignores Y. There's some methods that do something similar to PCA and pay attention to Y. There's a method called partial least squares, PLS, that is going to be summarize the X variables with the Y in mind. Also, there's the methods like LASSO that I talked about in the beginning. The feature selection methods; those are also going to pay attention to the Y variable. So, partial least squares, LASSO. Those are methods when you want to summarize the X data for a particular Y. They're going to be more useful for that particular scenario. Yeah, I think we mentioned most of that. Okay, so some key points from today. Global versus local methods. What are the difference between those? Global methods preserve the overall variance structure, can be used in regression because we can map them back. Again, if we have this party, we're trying to explain personality types maybe. We measure introvertedness maybe, and we measure sense of humor, and then we can plot people by those two variables. Maybe some groups come up there, and when a new person comes in, we can plot them. We can predict for them. The local methods that we didn't talk about today, they're more like, all right, let's have these people just get with people that they're friends with, and that will create some groups. They will preserve neighborhood structure. So variables that are similar initially are going to be, or excuse me, observations that are similar are going to be similar in these local methods. More for visualization only. Don't really use these in regression methods. The PCAs, some of the properties of them, they're uncorrelated. PCA1 has a correlation exactly equal to 0 with PCA2, 3, 4, so on and so forth. And they are ordered to explain in how much variance they explain. So PCA1 is going to explain the most, PCA2 the second most. So by design, that's always going to happen. How we choose the number of PCAs, we can look at the eigenvalues and the proportion of variance explained. Use the elbow, or say, all right, we want to explain 80% of the variance in the X data. The loadings, these are important to look at so we can interpret the components that we have. Our example with the food data, we saw that the first principal component separated things into basically foods that are from things that are alive and foods that are from plants. When we are implementing PCA, always scale the variables first, check for outliers. Sometimes we may want to transform the variables to make them have a more bell-shaped normal distribution. When we do principal component regression, we replace the original data with the uncorrelated principal component scores and regress Y on those scores. This eliminates collinearity. We do have to choose the number of PCs. We can do that by cross-validation, for example. In the example we did, we predicted systolic blood pressure using the PCA scores with linear regression. Principal component regression can be we can do it for logistic regression as well. So if we had a binary outcome, we could predict it using the principal component scores that we have. It could be a survival outcome. Okay, time to event. We could say, all right, well, is any of these principal component scores related to some time to event outcome? Doing the same thing we did here. You do a principal component analysis, put those scores back into the model, run the model as you normally would. With the last thing, connecting this back into genomics, for the previous days, they were doing some principal component analysis. We could do it here. We could look at the genomics data, do some principal component analysis. It's the same thing that we had above. It's just on a much larger scale. We did some principal component analysis, particularly on day 13, where we did the variance stabilizing transformation. Then we can do principal component scores on that. And then we could say, do some regression analysis for some outcome. I mean, for the genomics data, we only had 8 subjects, but yeah, we could look at the first 3 principal component scores and see which one of those principal component scores was related to treatment, for example. I think it'd be very clear that principal component 1 would be highly related to the treatment variable that we have. Great. We still have about 15 minutes left here, so why don't we start on with taking a look at the lab for today. We may as well. I'm going to open this up as a .qmd file, and in terms of the HTML. So the data set we're going to use here is a little different than normal. So these are chemical measurements on 178 wines from 3 cultivators grown in the same region of Italy. Each wine has 13 continuous measurements, including alcohol content, acidity, phenolic compounds, color intensity, and then we have some other chemical properties. Data set doesn't have any missing values. The variables are on different scales, so we will have to scale them. And the 3 cultivators give you a natural grouping to evaluate the PCA results without using that grouping during the PCA itself. So we'll apply the PCA and we'll take a look. Does this natural grouping that we get from the cultivators of the wine, does it correspond to what we find? The package, so if you don't have this, load this package up. Setting it up here, looking at the wines that we have and all the measurements from the wines. We have the soil here, and we have the label, which is the cultivator of the wine. Yeah. So yeah, so I forgot about this. So the data using the factMine-R, the wine data there has a little smaller structure. So you can use that one if you like. This one only has 21 lines and has 31 measurements. There's another one available at the using the UCI. This is the UCI is something that is a website that has a lot of different machine learning data sets there. So if you want to load the UCI one in, let's take a look at the UCI version. Yeah, so that wasn't too hard to do. Okay, so we have the cultivator here, just 1, 2, and 3, and then here we have all the chemical measurements available to us. And this data set is the one I initially described. So this one is just a little easier to get because it's already in this factominr package. But this one has a little larger sample size, so it's a good one to use. So this is a little look at the factominr version of the package. The one that I just loaded in is going to be a little different. We have some summary statistics for that. So here where we are creating the X variable, only keeping those variables that are numeric. So this one is going to remove that first variable, the variables that are character variables. So here, yeah, so we've made this cultivator a factor for this one. So that is not going to be a so that's not going to be a numeric variable. So that I just wanted to make sure that will work for either version of this data set. So I'm going to call this one line. And then do some cleaning on this version. Get some summary statistics. And we can run PCA. Good. So it works on both of these. Okay, this is a quick scree plot. Again, this is for the factomin version of the data set. We may have something a little bit different for the one that we download. Yeah, okay. Well, I'll leave that for the question of how many principal components to keep, but I think seems to be a good clean answer there. This is a scree plot of the principal component scores for dimensions 1 and 2. Group these by the different regions. So the exercises to compute the proportion of variance explained for all the different principal components. How many PCAs do we need to explain at least 70% of the variance, or at least 90? The Kaiser's rule, we keep PCAs with an eigenvalue greater than 1. We can get the eigenvalues, we're already using them in this variance explain. It's just the standard deviation squared. So if we use Kaiser's rule, how many should we suggest? And lastly, make a scree plot using the visualization. Say, all right, does there appear to be an elbow? Does it agree with the plots we get from B and C? Exercises 2, here we're going to produce a score plot of the PCAs like we did in the morning. Do the 3 cultivators separate into these first 2 PCs? Yes or no? PCA runs right to left. Look at where each cultivator falls along PCA. So does it seem like some cultivators are higher on PCA 1 versus others? You can make a second scree plot using PCA1 and PCA3 instead of PCA1 and 2. We just have to change the axis here. Does this improve visualization? Do we see more separation between the cultivators here? And then lastly, we're going to take a look at the loadings here. Similar to how we did in the morning, what variables have the largest loadings, positive or negative, for at least PCA 1 and 2, and then the biplot that we talked about. So let's focus on those first three exercises. None of these have to do principal component regression, but we're just going to focus on the principal component analysis piece for the lab. So you can get started on that. As far as which one to use, you can use either the facto-mind version of the wine data set or the one that I just showed that you can download. Either one is fine with me. They will likely give you different results, but should be similar to one another.